Dear students, last time we had seen the placenta and their types. Now we go in detail. The next topic is stem cells. Now, what are these stem cells? Stem cells are biological cells found in all multicellular organisms that can divide and differentiate into diverse specialized cell types and can self-renew to produce more stem cells in mammals. There are two broad types of stem cells: embryonic stem cell, which are isolated from the inner cell mass of blastocyst, and adult stem cells, which are found in various tissues. So, this is the introduction of the stem cell, but actually, we have to study the stem cell means during the development of the embryo. We know that there is a fusion of male and female gamete, zygote is developed, and that zygote divide through the cleavage, and the bunch of cells are formed called as morula or blastula, and these cells in future in the embryo differentiate into various cells, organs, and system. For example, digestive system, reproductive system, excretory system, like that. So all these organs, system, tissues are formed from the one cell or the bunch of these cells. So these cells of blastocyst are called as stem cells. So these stem cells nowadays is very important because in the developing embryo, stem cells differentiate into all specialized cells, and these cells are called as pluripotent cell. So these cells in future is very useful to cure some diseases. Now, where we get these stem cells? So there are three accessible sources are there. That first one is the bone marrow, then adipose cell, that is lipid cells, and third is the blood, which requires extraction through the pheresis, wherein blood is drawn from the donor. So now we see the properties of the stem cell. The classical definition of stem cell requires that it possesses two properties. Keep in mind, this is very important. Self-renewal, the ability to go through numerous cycles of cell division while maintaining the undifferentiated state. This is self-renewal. Second one, that is potency. The capacity to differentiate into specialized cell types in the strictest sense. This requires stem cells to be either totipotent or pluripotent to be able to give rise to any mature cell type. Although multipotent or unipotent progenitor cells are sometimes referred to as a stem cell. Now, potency here actually it can differentiate into any organ, specialized cell. Naturally, during the developing embryo, one cell is responsible for the organ or the system in the future embryo. That means they have that much of capacity or nucleotide sequence. Now, the self renewal means two mechanisms exist. Obligatory asymmetric replication, a stem cell divide into one father cell and is identical to the original stem cell, and another daughter cell is differentiated. Second one, stochastic differentiation. When one stem cell develops into two differentiated daughter cells, another stem cell undergoes mitosis and produces two stem cells identical to the original. So this is about the self renewal. Then potency definitions. Potency specifies the differentiation potential of the stem cell. Totipotent. Stem cell can differentiate into embryonic and extraembryonic cell types. Pluripotents are descendants of totipotent cells and can differentiate into nearly all cells in the future organ. Multipotent stem cells can differentiate into number of cells, but only those of closely related family cells. Oligopotent stem cells can differentiate into only a few cells. Then unipotent cells can produce only one cell type, their own, but have the property of self-renewal which distinguishes them from non-stem cells. So these are actually a capacity of particular stem cells to divide or to get the cells. Now see the diagram. By observing this diagram, you can very well understood. So what happened during this? See the zygote. That is totipotent. Now zygote we get the blastocyst. We studied all these things, so it is very easy to understand. So epiblast, that is the after the blastula stage, we are having the bunch of cells. Epiblast stem cells. If we remove the cells, this is called as epiblast stem cells. Then from epiblast we get the embryo. And from embryo, the germ cells are we get, and here the next stage is the adult one. Now the adult stem cells we get from the skin, central nervous system, bone marrow and other parts, and induced pluripotent stem cells for the use. So these are the kind of stem cells. Now by observing picture you will very well know. So identification, this is the identification of the stem cells. Now we get the idea about this embryonic stem cell first of all. So embryonic stem cell lines are cultures of cells derived from the epiblast tissue. See here, this is the epiblast tissue, embryonic stem cells. A blastocyst is an early stage embryo approximately 4 to 5 days old in humans and consisting of 50 to 150 cells. These are called as embryonic stem cell. Then the fetal stem cell. The primitive stem cells located in the organs of fetuses are referred to as fetal stem cells. Means in the organs of fetus. Then adult stem cells, also known as stem cells and germline, and they can be found in children as well as in adult. Pluripotent multi-adult stem cells are rare and generally small in number but can be found in a number of tissues, including umbilical cord, blood. Okay. Now you will get the idea. See here. Single cell embryo, 5 to 7 days embryo, infant and the adult. So here from all these stages we get the stem cell. Now this is too much long lectures. So we have to say next one, that is the amniotic stem cells. Amnion. What is amnion? Amnion, it is a extra embryonic membrane. We studied in the cheek embryology. So here multipotent stem cells are also found in amniotic fluid. These stem cells are very active, expand extensively without fetus and are not tumorigenic. And amniotic stem cells are multipotent and can be differentiated in cells of adipogenic, osteogenic, myogenic, endothelial, hepatic and also neuronal lines. That means what? That amniotic cells are the very basic cells. In the future, they can be produced. Just I have discussed the any one organ system in the adult body. Means osteogenic means responsible for the skeletal system. Myogenic means muscles. Endothelial means the endothelium tissues, organs. Hepatic means liver. Also, neuronal lining means brain. So it can be a very organ in the future. So that is developed from the amniotic stem cells. Then cord blood stem cells. So this cord blood stem cells is multipotent and displays embryonic and hematopoietic characteristics. So they can give rise to embryonic layer derived cells in presence of different inducers. Then induced pluripotent cells. These are not adult stem cells but rather adult cells and reprogrammed to give rise to the pluripotent capabilities using genetic reprogramming with protein transcription factors. Then, the lineage, stem line cells. Here, to ensure self renewal, stem cells undergo two types of cell division. Symmetric division gives rise to identical daughter cells, both endowed with stem cell properties, and asymmetric division, on the other hand, produces only one stem cell and a progenitor cells with limited self renewal potential. So, during this cell division, we are able to give the various organs in the developing embryo. And now, the most important part, that so how these are the stem cells in medical treatment. So we know that once again I'm summarizing this point. So stem cells are the basic cells in the developing embryo from the zygote, and these stem cells can be obtained from the blastocyst, then this morula stage, then from the fetus and from the adult. But why there is a research in stem cell and how they are useful to the human being? The stem cells in medical treatment. So this is the most important application of the stem cells. Why? Because medical researchers believe that stem cell therapy has the potential to dramatically change the treatment of human disease. A number of adult stem cells therapies already exist, particularly bone marrow transplants that are used in the treatment of leukemia. In the future, medical researchers anticipate being able to use the technologies derived from the stem cell research to treat the wider variety of disease, including cancer, Parkinson's disease, spinal cord injuries, amyotrophic lateral sclerosis, multiple sclerosis, muscle damage, amongst the number of other impairment and conditions. See, what is this actually? We know that all these organs and systems are derived from the stem cell. So the nucleotide sequence which are present in the gene of the stem cells are same as in the cells. So if we put the stem cells, all these nucleotide sequences get repaired and the disease is cured. This is the use of stem cell in curing the diseases in the future. Now, however, there is still exists a great deal of social and scientific uncertainty surrounding stem cell research. So actually, this is not so easy. And one concern of treatment is the risk that transplanted stem cells could form tumors and become cancerous if the division continues uncontrollably. If we replace, for example, in the blood cancer cells, so if we replace the bone marrow cells, we are producing the blood cancer cells. If we transplant that bone marrow from that patient and apply the future the new one from the another patient, so that bone marrow cells produce the good blood and cancer become cured. But in case of muscle and other things, if we replace the cells, that can grow uncontrollably and produces the cancer, maybe the chances. And supporters of embryonic stem cell research argue that such research should be pursued because the resultant treatments could have significant medical potential. And it has been proposed that surplus embryos created for in vitro fertilization could be donated with consent and used for research. So, this is the difficulty in research. So, this is actually a new topic for you people. You have to read 4 or 5 times and then you understand what are the stem cells. So, next time we will see the next topic, that is the biological clock. Thank you.